Integral membrane proteins, also known as intrinsic proteins, are proteins which are permanently anchored in the phospholipid bilayer via interactions between the protein's hydrophobic domains and the fatty acyl groups of the membrane phospholipids. These proteins have one or more segments that are embedded in the membrane and often span the membrane, known as transmembrane proteins. However, not all integral membrane proteins are transmembrane proteins. Peripheral membrane proteins are proteins which are only temporarily attached to the membrane via attachment to either integral membrane proteins or via non-covalent interactions with the membrane. There are two classes of integral membrane proteins. The first, integral polytopic proteins, also called transmembrane proteins, and the second, integral monotopic proteins. Within this group, there are two further types of protein, single-pass and multi-pass membrane proteins. Single-pass membrane proteins are those which pass molecules through the membrane just once, whereas multi-pass membrane proteins pass molecules through the membrane multiple times. There are 6 types of transmembrane proteins. Type 1 is where the carboxyl terminus is towards the cytosol. Type 2 is where their amino acid terminus is towards the cytosol. Type 3 is proteins with multiple transmembrane domains in a single polypeptide. Type 4 is where proteins with several different polypeptides assemble together. Type 5 is where proteins are anchored covalently to the lipid bilayer. And type 6 is proteins with both transmembrane domains and covalently linked lipid anchors. Integral monotopic proteins are proteins which are associated with one side of the membrane rather than spanning the entirety of it. There are 4 types of integral monotopic proteins. The first is an interaction by an amphipathic alpha helix parallel to the membrane plane, called an in-plane membrane helix. The second is interaction by a hydrophobic loop. The third is an interaction by a covalently bound membrane lipid. This is called lipidation. The fourth is by electrostatic or ionic interactions with membrane lipids, for example, through a calcium ion. Most integral membrane proteins are made up of a single alpha helix. These alpha helices are composed of roughly 20 non-polar amino acids which bend, maximizing the number of hydrogen bonds, forming a highly stable configuration. Single alpha helices are usually seen in recognition and receptors. Some integral membrane proteins are formed of helical bundles. These structures are mainly observed in enzymes, transporters, and some receptors. Another type of structure seen in membrane proteins is beta barrels. These have between 8 and 22 strands of approximately 10 amino acids, where every other amino acid is hydrophobic. The beta barrel structure fills the hydrogen bond requirements, forming a stable structure. These structures are observed mainly in membrane channels and pores. The location of an integral membrane protein's domain gives an indication of its function. Domains located on the extracellular membrane surface are usually involved in cell-to-cell recognition and interaction, or cell signaling. Those within the membrane are often channels or pores, and those on the intracellular membrane have a wide range of functions, including cytoskeletal anchorage and transduction of intracellular signaling pathways. Due to their hydrophobic membrane domains, integral membrane proteins can only be isolated using detergents such as STS, an ionic detergent, or Triton X100, a non-ionic detergent. This makes studying the structure of these proteins very challenging. In fact, it is estimated that fewer than 1% of the known high-resolution protein structures are of integral membrane proteins. Wow. This is despite them making up 30% of the proteins in a cell and over 50% of all current drug targets. Integral membrane proteins perform a wide range of functions. Some of these include intercellular communication, energy production, transport of material across membranes, cell-to-cell recognition, anchorage and attachment, signal transduction, and membrane fusion. We will now take you through examples of each of these to show you how structure informs function, and to give you an idea of the diversity that exists within this class of protein. A translocase is a channel protein which transports proteins into the endoplasmic reticulum lumen and the endoplasmic reticulum membrane. A translocase is a heterotrimeric protein. The largest subunit of the translocase is the alpha subunit, called Sec61 alpha in mammals, Sec61P in S. cerevisiae, and sec-Y in eubacteria and archaea. The beta subunit, called sec61-beta in mammals, SBH1P in S. cerevisiae, sec-G in eubacteria, and sec-beta in archaea. In eukaryotes and archaea, the beta subunit spans the membrane once, with the N-terminus in the cytosol. In eubacteria, the beta subunit spans the membrane twice. The gamma subunit, called sec61-gamma in mammals, SSS1P in S. cerevisiae, and sec-E in eubacteria and archaea. In most species, the gamma subunit is a single spanning protein with the N-terminus in the cytosol, but in some eubacteria, for example in E. coli, there is an additional transmembrane segment on the N-terminus which is non-essential to function. The pore of the translocase is contained within a single subunit and is an hourglass shape, with the smallest point of the pore being made up of 6 hydrophobic amino acids, which in many species are isoleucine. The pore is 5 to 8 angstroms in diameter and is flexible to allow bulky side chains to pass through. The pore is blocked by a small helix called the plug, which is the transmembrane segment 2A. The plug moves a distance of roughly 20 angstroms to a cavity on the extracellular side of the translocase to allow a polypeptide to move through the pore. The plug is present to stop the leak of ions between the cytosol and the endoplasmic reticulum lumen. The translocase works by binding a ribosome which is synthesizing a polypeptide and recognizing a signal sequence on the newly synthesized polypeptide, which forms a helix of roughly 2 terms, which intercalates between the 2nd and 7th transmembrane segment. The polypeptide sequence then moves through the translocase into the endoplasmic reticulum lumen for further processing, or the polypeptide will move through the translocase until a stop transfer sequence is in the pore of the translocase, at which point the translocase will open laterally to allow the polypeptide to move laterally into the membrane of the endoplasmic reticulum. There are 2 different ways in which polypeptides can move through the translocase: co-translational transport and post-translational transport. Co-translational transport is when the polypeptide is moving through the translocase as it is being synthesized by a ribosome. The energy for this comes from GTP hydrolysis in the polypeptide synthesis. Post-translational transport is when the polypeptide is transported after synthesis. This involves the use of a ratchet mechanism which uses a molecule called BIP. BIP, with bound ATP, has a binding pocket in which the polypeptide can fit. The ATP gets hydrolyzed, causing the binding pocket to close on the polypeptide. This pulls the polypeptide through the translocase. Another set of translocases are responsible for moving nuclear proteins into the mitochondria and chloroplasts. In mitochondria, there are 5 integral membrane protein complexes involved in this process: the TOM, TIM22, TIM23, SAM, and OXA1 complexes. These complexes all use a combination of beta barrels that act as pores and alpha helices that act as receptors that recognize specific target sequences to perform their functions of moving proteins into the different areas of a mitochondrion. For a protein to enter any area of a mitochondrion, it must first move through the TOM complex on the outer membrane. Beyond this, the SAM complex inserts proteins into the outer membrane, TIM22 inserts proteins into the inner membrane, TIM23 moves proteins into the matrix, and OXA1 facilitates protein movement from the matrix to the inner membrane, providing a second pathway for insertion. Chloroplast translocases are very similar. TOC and TIC complexes have similar structures and functions, however there is only 1 TIC compared to the 2 TIMs mitochondria possess. An example of intercellular joining are known as gap junctions. They are clusters of intercellular channels that allow direct diffusion of ions and small molecules between adjacent cells. Formed by head-to-head docking of hexameric assemblies of tetraspan integral proteins, the connexons in chordates. In prechordates, gap junctions are made up of inexins. These channels cluster into polymorphic plaques containing a few to thousands of units. This close membrane apposition, required to allow the docking between connexons, sterically excludes most other membrane proteins, leaving only a small 2 nanometer extracellular gap for which it is named. Most cells express multiple connexins which co-oligomerize into either homomeric, the same, or heteromeric, mixed connexons. These connexons can then dock to either an identical connexon, forming a homotypic intercellular channel, or with a different connexon, forming a heterotypic channel. Only some assembly combinations are permitted. However, there is still a large number of intercellular connections able to be formed. This leads to diverse functions of intercellular joining, as different combinations result in different physiological functions and properties. Enzyme activity. ATP synthase is highly conserved in cell membranes of bacteria, inner membrane of mitochondria, and thylakoid membranes of chloroplasts. The structure of ATP synthase. ATP synthase consists of two main regions, F1, named from factor 1, and FO, named after binding of oligomycin. The FO region, or the rotator subunit, is made of 12 C subunits. Protons enter through subunit A of the stator, causing it to rotate. Once rotated 360 degrees, the protons leave via a second proton channel in the stator. The rotor is responsible for using a proton gradient to convert electrochemical energy to mechanical kinetic energy. The central stalk, the gamma subunit, transfers energy from the FO rotatory region to the F1 ATPase catalytic region, responsible for ATP generation. The FO region and the shaft both rotate, but the F1, held in place by the peripheral stalk complex made of the A subunit, the stator, which is bound to the FO region, and B subunit, which is bound to the F1 region. As the central shaft rotates within the F1 region, conformational change of the catalytic subunits occurs. The catalytic subunit is made of 3 dimers of A and B subunits. Catalysis occurs at the interface between the dimers. Each dimer is made of 1 alpha and 1 beta subunit. The 3 interface steps are: ADP and PI bind. The central staff rotates 120 degrees to rearrange molecules. Further 120-degree rotation occurs, fusing ADP and PI to form ATP. A further 120-degree turn occurs to return to the starting position, where ATP is released and ADP and PI binds again. Overall, an ATP molecule is made from 3 protons crossing the membrane. One 360-degree rotation produces 1 molecule of NADH, 3 molecules of ATP, and 10 protons across the membrane. Furthermore, ATP hydrolysis shows that the direction of rotation can be reversed. The proof for rotation came from binding fluorescent actin to ATP synthase and visualizing it under a microscope. When ATP was added, the rotor starts spinning, showing the fluorescent filament spinning too. The proof for rotation came from binding a fluorescent actin filament to ATP synthase, and once ATP had been added, the actin was spinning clockwise. Therefore, the ATP synthase was spinning clockwise. For ATP hydrolysis, the ATP synthase spins clockwise. For ATP synthesis, the ATP synthase spins anticlockwise. There are 2 main forms of ATP synthase molecules: F-type, which is the mitochondrial ATP synthase, which uses energy from ATP synthesis to produce a proton gradient; V-type, which is lysosomal, which uses energy from ATP hydrolysis to produce a proton gradient. They have similar structures but rotate in opposite directions. Finally, the cristae shape of mitochondria is formed via dimerization of 2 ATP synthase molecules. This increases ATP synthesis, as this focuses proton gradients near ATP synthase. Located on the inner mitochondrial membrane, the electron transport chain comprises of 5 large protein complexes, known as complex 1 through 5. Its role is to produce ATP by passing electrons from complex to complex in a series of redox reactions via the formation of a proton gradient, ultimately passing electrons to complex 5, known as ATP synthase, which, as discussed earlier, produces the ATP. Complex 1 is an L-shaped protein which has one arm embedded in the inner membrane and the other extending into the matrix of the mitochondria. It is comprised of 45 varied polypeptide chains, including flavin mononucleotide and at least 8 enzymes containing iron-sulfur. It catalyzes 2 simultaneous and obligatory processes. First is an exergonic transfer of ubiquinone of a hydride ion from NADH and a proton from the matrix. The second is the endergonic transfer of 4 protons from the matrix to the intermembrane space. Overall, it acts as a proton pump to move protons from the matrix to the intermembrane space, which is responsible for 40% of proton motive force. There are 7 integral membrane protein subunits of the membrane arm embedded in the membrane, where 3 are responsible for pumping 3 protons, with the 4th subunit pumping a proton nearest to the Q-binding site. Complex 2 is the only membrane-bound enzyme in the citric acid cycle and forms a link between it and the electron transport chain. Complex 2 couples the oxidation of succinate at one site with the reduction of ubiquinone at another site. It contains 5 prosthetic groups of 2 types and 4 different protein subunits, as well as a heme group which suffices to reduce electron leak, which is a cause of the production of reactive oxygen species. Complex 3 couples the transfer of electrons from ubiquinone to cytochrome C. Its functional unit is a dimer, where each monomer consists of 3 proteins central to the action of the complex, known as cytochrome B, C, and the Risk iron sulfur protein. 2 cytochrome B monomers surround a cavern in the middle of the membrane, where ubiquinone is free to move from the matrix side to the integral membrane side, which effectively shuttles electrons and protons across the inner mitochondrial membrane. Complex 4 transfers electrons from cytochrome C to oxygen. It is a large dimeric enzyme, where each monomer contains 13 subunits. In bacteria, complex 4 exists in a simpler form of only 3 or 4 subunits. This suggests that these 3 subunits are highly conserved in evolution, whilst the other 10 subunits may contribute to the assembly or stability of complex 4. Subunit 2 of complex 4 contains 2 copper ions complexed with the sulfur groups of 2 cysteine residues in a binuclear center. Subunit 1 contains 2 heme groups and a copper ion. In a laboratory setting, these complexes separate into 5 distinct complexes. However, in the intact mitochondrion, they exist in tight association with each other. Kinetic evidence favors electron flow through a solid state, which favors the respirazone model, which is the complexes associating into larger supercomplexes. Photosystems are composed of 2 main compartments, the light harvesting complexes and the reaction center complex. The outer region of photosystems consists of light harvesting complexes, which are composed of an association of proteins called CP43 and CP47, and pigment molecules like chlorophylls and carotenoids. The inner core of photosystems comprise of the reaction center complex. The main components of the reaction center complex consists of a pair of chlorophyll A molecules, a primary electron donor, P680 or P700, and a primary electron acceptor. The role of the light harvesting complex is to transfer light energy from photons in a process known as resonance energy transfer from chlorophyll molecules in the light harvesting complex to the chlorophyll A pair in the reaction center complex. After the chlorophyll A pair, electrons are transferred to P680 or P700, tetramers of equally spaced chlorophyll A molecules. After P680 or P700 electron flow is unidirectional through the primary electron acceptor, pheophytin for photosystem 2, or iron-sulfur cluster proteins for photosystem 1. The P for P680 and P700 stands for pigment, and the following number indicates the peak of the absorption spectrum in nanometers. The reaction center complex's functions are different between photosystem 1 and photosystem 2. Photosystem 2 transfers electrons from water to plastoquinone. Cytochrome Bf links the 2 photosystems by transferring electrons from the product of photosystem 2 to the reactant of photosystem 1, which also generates a proton gradient. Photosystem 1 uses light energy to generate reduced ferredoxin, a powerful reductant. Ferredoxin NAD+ reductase, a flavor protein, uses 2 ferredoxin molecules to convert NADP+ into NADPH. Ferredoxin, despite being a strong reducing agent, only carries 1 available electron and therefore is not useful in driving many biosynthetic reactions. NADPH, a versatile 2-electron reductant, is much more widely used in metabolism, including reactions in the Calvin cycle. Glycoproteins are a ground substance in extracellular matrices and classed as proteins which have attached carbohydrate groups. Sugars either form an N linkage to the amide nitrogen atom in the side chain of asparagine, or an O linkage to the oxygen atom in the side chain of serine or threonine. Asparagine residues can also accept oligosaccharides if followed by a serine or threonine residue. Fibronectins act as a biological glue. They adhere cells to the extracellular matrix. Fibronectin fibers form pathways for migrating cells, which is especially important for development, organ shaping, and wound healing. They are flexible and composed of non-identical dimers linked by two sulfur bridges at the C-terminal and are synthesized by many cell types. There are two forms, soluble in plasma for blood clotting and insoluble fiber in the extracellular matrix, and contain multiple specific binding domains. Fibronectins are the most abundant multi-adhesive protein. Laminins are important in basement membranes. They are made of a number of alpha, beta, and gamma chains. The N-terminus interacts with other extracellular matrix proteins, which assemble and improve stability of basement membranes. The C-terminus interacts with cell surface receptors for a diverse range of functions including adhesion, migration, apoptosis, differentiation, and gene expression. Fibrillin. Molecules of fibrillin 1 attach to each other and to other proteins to form thread-like filaments called microfibrils. Sibling, small integrin-binding ligand N-linked glycoprotein, play a role of mineralization of bone and dentine. Protein glycosylation occurs in the lumen of the endoplasmic reticulum and inside the Golgi. N-linked glycoproteins acquire their initial sugars from dolichal donors in the endoplasmic reticulum. Proteins are then transported to the Golgi complex via transport vesicles for further glycosylation and sorting. An example of integral membrane proteins that are involved in signal transduction within a cell is the G protein coupled receptors, called GPCRs. These are receptors which, when activated by ligand binding, undergo a conformational change which allows the binding of a G protein, which can in turn be activated via a conformational change which causes GDP to dissociate and GTP to bind. Is that the transmembrane domain is made up of 7 alpha helices that are arranged to form a barrel-like cavity which can bind ligands. This snake-like structure that weaves through the membrane is responsible for GPCR's alternative name, serpentine receptors. GPCRs also have extracellular N-termini and cytoplasmic C-termini which interact with ligands and G proteins respectively. There are 6 classes of GPCR, classes A to F. Class A is the only family which binds ligands within the transmembrane domain. Classes B to F have varying N-termini which bind ligands in different ways, but which all ultimately activate the receptor to produce a signal. When membranes are targeted to each other, they need a way to fuse together. In order to fuse, membranes need to be within 1.5 nanometers of each other. This is mediated by proteins called SNAREs. There are 2 types of SNARE protein, V SNAREs, which are located on the vesicle, and T SNAREs, which are located on the target membrane. SNARE proteins are 100 to 300 residues long and have a conserved SNARE motif of 60 to 70 residues, which forms a complex with another SNARE motif. There are 36 SNAREs in humans. 31 of those SNAREs have membrane anchors at the C-terminus. Others, for example SNAP25, have a membrane anchor in the middle. The vesicle that is being targeted to the membrane is covered in V SNAREs, but the interaction between SNAREs is 1 V SNARE to 3 T SNAREs. The 3 T SNAREs can be 3 separate T SNARE proteins, or can be 2 T SNARE proteins, one of which has 2 SNARE motifs. A T SNARE with 2 motifs has one at the C-terminus and one at the N-terminus, with the membrane anchor in the middle, whereas a SNARE with 1 motif has the motif at the N-terminus. The T SNAREs are made up of 1 heavy chain and 2 light chains. The heavy chain only has 1 SNARE motif, and the light chain can have 2 SNARE motifs, or there can be 2 light chains with 1 SNARE motif each. When two membranes are brought close together by the machinery that is moving the vesicle, the snares wrap up together in an autocatalytic process to form a 4-helix bundle called a trans-snare complex. This complex is cylindrical and roughly 120 angstroms in length. The wrapping of the snares causes the membranes to be pulled together and forces water out of the gap between the membranes. The snares pulling on the membranes causes the membranes to become distorted so that the membranes can fuse easier. Once the membranes have fused, the snare complex becomes a cis-snare complex, which just means that they are on the same membrane. The cis-snare complex has no use to the cell, so it needs to be broken down. This is done by a hexameric ATPase called NSF and 3 proteins called SNAP. This forms the 20S complex. The NSF hydrolyzes ATP to provide energy to allow the snares to be twisted apart. V snares and T snares have been found to work in each other's place in some cases, so this has led to the new theory of Q snares and R snares. The name is determined by whether the residue in the middle of the snare is an arginine, R, or a glutamine, Q. In this theory, the R snare acts as a V snare and the Q snare acts as a T snare. Integrin is a transmembrane protein found only in animals involved in anchorage and cell signaling. Integrin links cells to the extracellular matrix, and the signals transmitted by integrins can influence differentiation, motility, growth, and survival. Integrin is a heterodimeric protein with an alpha and a beta subunit which are non-covalently linked. Extracellular ligands bind between the alpha and beta head groups. For example, the RGD sequence of fibronectin. Integrins bind their ligand with a lower affinity than cell surface receptors, but integrins are present with a 10 to 100-fold higher concentration. The integrins cluster together to form a dense plaque. There are 18 different alpha subunits and 8 different beta subunits. The subunits have a short cytosolic C-terminal domain and a large extracellular N-terminal domain. When integrin is in its inactive conformation, the external segments are folded together so that the receptor sites are not exposed, and the cytoplasmic tails are hooked together so that the binding sites on the beta subunit are not exposed. When integrin is in its active conformation, it has its external segments and cytoplasmic tails extended. Integrin can be activated by an outside-in or an inside-out mechanism. The outside-in mechanism works by a sequence, for example the RGD sequence, binding to the extracellular domain, which leads to an active conformation so that the cytosolic binding site of the beta subunit is exposed. The inside-out mechanism works by a molecule, for example the protein tailin, competing with the alpha subunit for its binding site on the beta subunit. This is mediated by signals that impact the ability of tailin to bind. Integrins link the cell to the extracellular matrix by binding actin and forming actin-linked cell matrix junctions, or by binding intermediate filaments to form hemidesmosomes. Integrin is influenced by tension. When integrins are under tension, they strengthen their grip on the molecules they are bound to, and when the tension is relaxed, integrin relaxes its grip on the molecule it is bound to. Integrins also have a role in cells which are required to be linked to the extracellular matrix in order to grow. This is called anchorage dependence. Mutations that can override anchorage dependence occur in cancer. Here is a list of extra reading for each of the topics presented.